Hi, my name is Francesca Lembeck. I'm a junior here at GCU, graduating in December this year with my bachelor's in biology with an emphasis in pre-medicine. And I would like to share with you the importance of studying ovarian cancer immunotherapy research using organoids. Just to give you a little background of how I came across this research and why it has become important to me, I had the fortunate opportunity to intern under Dr. Marion Curtis in her ovarian cancer immunotherapy research lab at the Mayo Clinic last year from January through July. Her lab focuses on immune cell metabolism, tumor antigens, and the tumor microenvironment of ovarian cancer and how that applies to the immunotherapy research. My project in her lab focused on the successful organoid growth containing both healthy and cancerous tissues so that we could test immunotherapy techniques on them. Although I cannot present any data that was obtained in her lab that is not yet published, I would like to share with you the importance of studying ovarian cancer immunotherapy options and how the use of organoids can be helpful in that area of cancer research. Ovarian cancer, which begins in the ovaries, is one of the most dangerous cancers because most cases, 76%, are not detected until it has progressed into stage 3 or 4, leading to a low survival and remission rate. Ovarian cancer metastasizes very quickly to other parts of the pelvic area, including the bladder, colon, rectum, and intestines, the abdomen, most commonly the omentum, and eventually to the lungs, liver, and lymph nodes. Ovarian cancer is composed of a diverse group of tumors. Most of these are high-grade serous carcinomas, which arise from the implantation of epithelium from the fallopian tube. Endometriosis, which is the growth of endometrial tumor in other areas of the pelvis outside of the uterus, is a precursor for endometrioid and clear cell tumors. Again, these metastasize to omentum, pelvis, abdomen, and then other tissues as well. There are two types of ovarian cancer, type 1 and type 2. Type 1 ovarian cancer is composed of one of 5 carcinomas. The low-grade serous carcinoma, which originates in the ovary or peritoneum, is rare, chemotherapy-resistant, and slow-growing. The low-grade endometrioid carcinoma starts in the endometrium or ovaries and is associated with endometriosis. This forms from mutations in the CTNNB1 or catenine beta 1 protein coding gene, P10, which is a phosphatase and tensin homolog mutation. This is a protein coding gene that is also a tumor suppressor protein, and the PIK3CA gene, which encodes a lipid kinase involved in multiple signaling pathways. Mucinous carcinoma begins in cells that line the ovary that produce mucin. CRAS mutations, which is a gene that acts as an on-off switch in cell signaling and controls cell proliferation, occurs in greater than 50% of these carcinomas. Transitional Brenner carcinoma arises directly from pluripotential surface epithelium, or stem cells, of the ovary, and form cells with urothelial potential, which are able to change shape and stretch. Clear cell carcinomas are malignant epithelial cells with a clear cytoplasm. This carcinoma occurs in less than 5% of ovarian cancers and is associated with endometriosis. The mutation involved in this is also the PIK3CA activation mutation. These carcinomas are genetically stable, and the KRAS, BRAF, and ERBB2 mutations occur in 2/3 of the cases. These originate from the ovarian mesothelium, resulting in an inclusion cyst which undergoes malignant transformation. This image is showing the development of low-grade endometrioid and clear cell carcinoma. The endometrial tissue implants on the ovarian surface, forming an endometriotic cyst, resulting in the development of the EMC or CCC cancer. Type 2 of ovarian cancer is highly aggressive and evolves rapidly. These include conventional high-grade serous carcinomas, which generate in the serous epithelial layer in the abdominal pelvic cavity, then found in the ovary. This has the lowest survival rate and is dangerous because it is typically asymptomatic until the advanced stages. Undifferentiated carcinoma is also rare, has a very low survival rate, and contains cells that are very immature. Malignant mixed mesodermal tumors is an endometrioid tumor containing both epithelial and mesenchymal components. This is also very rare. TP53 mutations are found in over 80% of these carcinomas. This gene codes for tumor protein 53, which is a tumor suppressor protein, which explains the rapid evolving of these carcinomas. These tumors rarely contain the same mutations as type 1, which is how they are able to differentiate between the two types. CCNE1 amplification is important to note because this codes for the protein cyclin E1 that results in the initiation of DNA synthesis. The amplification of this gene is critical in the survival of ovarian cancer. The majority of these carcinomas arise from a STIC, which is a serous tubal intraepithelial carcinoma, and is present in greater than 70% of high-grade serous carcinomas and is detected in the fimbria. Other development pathways would be a dislodgement of normal tubal epithelium from the fimbria, which is then implanted where the ovulation occurs, forming the inclusion cyst and developing into a malignant transformation. The images on the right, the top image is showing the proposed development of low-grade and high-grade serous carcinoma. Picture A is showing the normal tubal epithelium that is shed from the fimbria, forming the inclusion cyst on the ovary. The mutation depends on the grade of serous carcinoma. And picture B is showing the shedding of the STIC cells onto the ovary where the tumor mass is then formed. The bottom picture is showing the STIC, or the serous tubal intraepithelial carcinoma. Picture A on the left is showing the hematoxylin and eosin stain, and on the right it is showing the immunohistochemical stain for P53. The asterisks define the boundary of lesion. Organoids, or mini-organs, are tissue-specific and grown in vitro. They are helpful in tissue-specific disease studies and treatments. Because stem cells from the organ that you would like to target are used, organoids mimic the complexity that each individual organ possesses. One of the tissues that we wanted to grow into the organoid was the fallopian tube, and this has been successfully done in fallopian epithelial stem cells in mice, so that is what we did. It is important to mimic the tumor microenvironment that naturally occurs, so the best growth conditions are still being tested. There has been success in long-term growth of greater than 6 months. This image on the right is showing the different stages of the fallopian tube organoid growth. There are many different factors that we considered that have shown to be successful in an organoid model. Successful growth with arspondin-1, which is an activation protein for wild-type air-liquid interface intestinal organs, has been shown. Successful growth with WNT3A, which is a protein-coding gene, EGF, which is epidermal growth factor, noggin, a developmental protein, and RSPO1, again an activation protein, for tumor epithelial organoids in submerged matrigel, which is a growth medium. Human samples contained CD3 plus T cells close to the tumor epithelium, showing that they are trying to target the tumor cells. They also showed CD8 plus and CD4 plus T cells, B cells, natural killer cells, natural killer T cells, and infiltrating CD3 plus T cells expressing the PD-1 protein. The PD-1 protein is the programmed cell death protein, which is how the tumor will keep growing. It programs it sends this protein to program the death in these cells that would normally be trying to attack the tumor. Interleukin-2, preserved intraorganoid PDS, CD3+, tumor-infiltrating lymphocyte clusters, CD4+, and CD8+ subsets, but no longer than 60 days. and anti-PD-1 and anti-PD-L1 increased CD8 plus tumor-infiltrating lymphocytes. So it is important to add the anti-PD-1 and anti-PD-L1 proteins. By increasing the tumor-reactive T cell count in the tumor organoid, the T cells will recognize and attack the tumor cells, but will leave the healthy tissue behind. This can be done by adding PBMC to the organoid culture to increase the peripheral blood lymphocytes within the organoid. This method has previously been successful in patients with mismatch repair deficient colorectal cancer and non-small cell lung cancer. The continuation of this research is extremely important. Because of the extremely low percentage of early detection, ovarian cancer is usually very difficult to treat. Once it has metastasized, surgery and chemotherapy options are very risky and often only prolong life a few years at best. Immunotherapies directed towards those cancer cells can recognize, target, and attack the cancer cells throughout the body without compromising the healthy tissue. This treatment can increase the success rates by providing a safer and more effective way of treating this cancer, which is given in the form of a vaccine. Once developed, this can change not only the way that ovarian cancer is treated, but also how all cancers can be treated and targeted. Here are my references. Thank you all so much for your time and consideration and for listening to my presentation.